I want to move this little thing here. Okay, thanks for being there. I'm so excited to be the first one to give those kind of summary seminars that we were doing in ABERG. And as you know, I started with ABERG as an undergrad, and I was working closely with Miles Silman, Norma Salinas, and Jason Bradford, who we started to work in the transect. And this is a collaborative effort. I'm mainly working, with Ken Feely, Jeff Binder, also with Marbuch, Patrick, and Cezanne, and also with collaborations of the Rainforest people, because the transect just not end in the Costa Ñuñoa Valley, but that goes down to the Amazon. So here's a big question: how the Andean Amazonian forests are responding to global warming? And to answer that, first I want to ask, what sets a distribution? What sets a species distribution? And this is still a fundamental question in ecology. And in this talk, I will show you how the system is now, how it's changing. And those changes are community and species level, and also the implications in the ecosystem properties. So what says distribution, species distribution? Like I said, this is a really standing question in ecology, and the use of environmental gradients as natural laboratories to understand species distribution is old as the discipline of ecology itself. Here you are appreciating a fantastic illustration of Francisco de Caldas, where you can see how the ecosystems are changing from the lowlands to the highlands in the Andes. And, And we know that species are distributed along the environmental gradients, and there is like a first main axis that we know, it's temperature, precipitation, and soil nutrients. And here I will talk about temperature. We know that temperature is increasing over time. That's not novel, but what is important to remember is that as a mean global temperature surface, we already passed 1 degree Celsius. So more or less like in 100 years, since the 80s, temperature of the global surface increasing 1 degree Celsius. And the question is, how those tropical forests are responding to this global warming. And those are the places where we should concentrate our efforts to understanding this process and these responses, because we know that tropical forests are highly vulnerable to climate change, basically because we see species with narrow climatic ranges, and those systems are biodiversity hotspots with high endemism, and more importantly, because we depend on them. So today, for the people that know the transect and for the new people that haven't been there yet, I will show you how it is. So today, I will show you how doing these basic but important measurements in the forest, we can understand how those three communities are responding to this global change and how that affects the ecosystem function. So for this talk, first I will introduce the transect in Peru. I will show you some general trends along the gradient. Then we'll explore if those species are shifting their distributional ranges, basically looking at the shifts in species composition, changes in the relative abundance, process called thermophilization. And also we'll see how those changes in tree demography and species distribution are affecting the ecosystem function. So we'll be more focusing in carbon dynamics. So the transect, it's located in the southeastern Peru that goes up from the tree line to the lowlands, where you see this little square. This is a map of digital elevation. And just to reinforce the idea of this, is like you sitting in Panama and looking at Alaska at the same time. So this is to put you in the perspective, how long is the transect? This is our study area. This is the backbone of our study. So this is the transect in Manu National Park. You see that kind of brownish color is the transition between the forest and the grasslands. And then that ridge that you see, that's the actual elevational gradient that we use as a natural laboratory to study the effects of climate change in these tropical ecosystems. Each square that you see are those permanent plots. Each one is 1 hectare. And you see we have this beautiful gradient in elevation, but also we have this gradient in temperature as well. This is how the plots are distributed in the landscape. So each dot represents 1 permanent plot. This is the network that we started in 2003. But to have a more comprehensive understanding of this process of responding to climate change, we add these lowland plots working with Rainfor network and also with GAM. So Rainfor led by Oliver Phillips and Jadvinder, and GEM is led by Jadvinder Mali. So we include all the plots from the lowlands to the Andes, and we have 41 1-hectare plots with multiple measurements, and the gradient goes from 190 meters in the Amazon basin to 3,700 meters in the tree line. And we have this temperature to between 8 to 26 degrees Celsius in a really short geographic distance of 270 kilometers. And this work is only possible with a teamwork because this is not a one-man job, and mainly acknowledge all the hard work of these Cusqueñan students that spend a lot of time with me in the field during these multiple field campaigns. So in each of the census, like I mentioned before, we have those plots with multiple census over time. So we tag, ID, and remeasure every stem in each plot. And after 2 to 5 years, we remeasure the same stem, marking and making sure that we include all the stems above 10 centimeters of DBH, which is the minimum size of the trees that we measure in those plots. And working in the field has many challenges. So in the Andes, it's hard to find first where is the floor and to find where is the optimal point of measurement. So to install those plots and remeasure every time, we follow like standardized protocols so we can compare those results. So the trees in the Andes, they don't grow straight, so they grow like a J shape. So it's fun where to find it's fun to find where is the base of the stem and then trying to actually find this point of measurement. It's a little hard because you basically are walking over dead trees in the Andes. And in the lowlands, the trees are straight and it seems easy to work, but they have their own challenges too, like these big root structures where you have to be careful where to measure, and you have to do sometimes these human towers to have better measurements of the DBH. And we work too in the floodplain areas where you basically have to sort these challenges to measure those plots. And in the transect, just want to remind you that we start from scratch. So we didn't have any really good knowledge about even basic understanding of how was the floristic. So we start collecting all plant specimens and following all these standardized protocols for this baryon preservation, collecting all those samples and sending to different herbarium in Peru, Bolivia, and United States. And as a surprise, we find some new species, which is really interesting and exciting to find new trees in the transect. So there are many of them now. But some people related to the botany will say, when you go to this really unexplored sites, there is more likely to find a new species. But what about a new genus? And this is what we found, a new genus. It's a canopy tree, over 20 meters high, and the diameter is over 50 centimeters. It's a beautiful, abundant tree that it's distributed as well, not just in Manu, but we found some populations in Central Peruvian Oxapampa, Cordillera del Condor. And this was a newly collected tree genus, not just a hiding genus that were like stored in the herbariums for many, many years. So the name is Incadendron, and is naming after the Inca Empire because it fits their distribution more or less in the Andes. And the specific epithet is Esseri, which is dedicated to the specialists of this tribe of species. Sorry. And when we found this new genus, it brought the attention of the media from local and international, because they were really excited to know that we found this new genus. And you can see this media like in English, in Spanish, and even in French, talking about this new discovery. So now I will show you more or less about the climb in the transect. So in the x-axis we'll have elevation, and the y-axis precipitation. In this case is this black line. As you see, there is an increase in precipitation around, from the Amazon to mid elevations, and almost reach like 8 meters per year. And then that decreases to the tree line. So even in the tree line, we have almost 2 meters of rain, which is really humid system. From temperature, the red line, it correlates with elevation. So it's highly correlated with elevation. So this is why when we talk about elevation, we're basically talking about temperature. This blue bar that you see, it's the cloud base, which is when the clouds are rising up, that is where the point, there's this persistent zone where the cloud is in the transect. And it's around 1,500 to 2,000 meters. And then after that, we have this cloud immersion zone where you have this persistent clouds over the time and all year. And after that, we have this green bar, which is the tree line. And one thing to mention that only happens in the Andes or in the mountain systems, it's this landslides where can change the demography of the trees in a really short times. And there is a nice work from Catherine Clark about the landslides. And Cassie Freud, PhD students from Miles, is continuing this study looking how that affects the ecosystem function too. So stay tuned for those results. This is the inventory data. So we have 30 years of plot data, including the lowlands and the Andes, or the Amazon and the Andes. And each plot were census every 2 to 4 years. And all the stems of tree ferns, palms, and lianas were included. So we have over 42,000 individuals and around 1,900 species. So it's really highly diverse system. I will show you some of these basic trends in the transect to understand how the forest is. So first here we have elevation in the x-axis and then species richness. So each dot represents each permanent plot. And this dashed line that you see it's kind of this arbitrary division between the Amazon and the Andes that the people draw. And I put it here as a reference. The blue bar that you see is the cloud base, and the red and blue colors are the means or average of species per plot for the Andes and Amazon. So that what you see here is that from the Amazon, species richness, it's kind of constant until the cloud base and then decreases linearly to the tree line. So this is saying that the Amazonian forest can be as rich as the Andean forest, especially in the lower part, and then decreases. And we know that in tree line, tree diversity is really low. And then if we look for mortality, so each dot here represents a one plot again, and the size of the circle represents the number of censuses. So we have more censuses in the lowlands. So this is mortality rate per year, per hectare. So what we see here is that from the lowlands, it increases around the cloud base and then decreases to the tree line. So there is not a linear relationship. And overall, there is great mortality rates of 2.3% per year. And we see that in the Andes, the rates as a mean are greater than the Amazon. Well, slightly higher, we'll say, but still. But the main point here is to see that around the cloud base, it's where a lot of trees are dying. And this is important, just keep it in mind for later on. And this is the recruitment rate along the elevation. So we were expecting to have this linear relationship with elevation, but seems that you see here is kind of flat and then decreases to the tree line. We were expecting to have more in the Amazon because trees grows faster in the Amazon forest. And those rates were, again, positive, but more or higher in the Amazon than the Andes as we were expecting as average. And this is the net change in the stem numbers. So basically, blue colors represent positive change and red colors negative. So it means that the plots are losing or gaining more individuals per year. So the net change, again, around the cloud base, it's negative and really low, meaning or suggesting that those plots around the cloud base are losing more individuals that are gaining more individuals. And the mean for the whole transect is negative. So they are losing more stems that are gaining more strength, so meaning that mortality rate is greater than recruitment. And if we compare just the Andes and the Amazon, we see that in the Amazon, those rates are positive, and in the Andes is negative as a mean. So again, suggesting that the Andean plots are losing more individuals that are gaining more individuals. So understanding these trends in the demography, then we can see if those species are changing the distributional ranges, and we will explore this thermophilization process. But before, just to send you in mind that there is a paleo evidence of species migration, right? And this is a nice study led by Bush and also where Miles and Dunya work. And I don't know if I will do a good job explaining this, but maybe we can ask later to Mark. So here, basically, you see this reconstruction of temperature and elevation based on pollen records. The blue colors means zero probability of taxa, and the warm colors to the red means high probability of taxa. So you see that over time in the x-axis, so there is a constant in the taxa, but then you see this abrupt transition here, just in the transition between the Pleistocene and Holocene, and there was an increase of 1 degree Celsius per millennium. And after that, then you see this movement of species of the taxa, suggesting that there was a species migration. So here we see that temperature has increased in 1 degree Celsius over a millennia, but now we are observing a temperature increase of 1 degree Celsius more or less in 100 years. So that's a challenging for this forest, how they will be responding to these novel environments. And if you want to see how to prove that in the field, you can just go to the Andes. This is in Cusco, around 4,000 meters. So I'm hugging a legume. This is a fossilized tree. And that was alive like 20 or 30 million years ago. And the elevation was around 2,000 meters, but now it's 4,000 meters. So you can go to those sites and see those fossils. In the mountain systems, the species have narrow elevational gradients, and we can observe here in the figure. And this is an example of these species with narrow elevational ranges. We have here Ilex siciflora. So there is, this, that you see these dashed red colors. So this species is distributed from Bolivia to Ecuador, but it has just this narrow range of 500 meters, more or less, which is equivalent to 2.7 degrees Celsius. And over time, we know that those gradients of the ranges are not static because they're moving. So those ranges can shift upslope or downslope, or they can increase the range or they can contract the range responding to the environmental gradients. And those changes will have impact in forest and ecosystem properties. In the transect, we know that temperature has been increasing at the rate of 0.03 to 0.05 degrees Celsius per year since the 50s. And it's predicted that will increase over 4 degrees Celsius at the end of the century. So the question now is that, are those species shifting their distributional ranges? If so, can we observe those? To answer that, we will see this thermophilization, or we will test the thermophilization hypothesis, which is at the community level, which basically says is this wisdom of the crowd. I'll explain later how this process is, where all the species in the community are responding in different ways. And then the migration hypothesis, which is based on the taxon, is how the populations of each species are shifting the distribution. So how's this thermophilization process? So species are under the climate change, and species are shifting their distribution. And this climate change is favoring to these warm adapted species, where they will increase their abundance, but they don't not favor to the cold adapted species, because they will be reducing their number of individuals, their abundance. So under this thermophilization process, the more adapted species are going to increase in abundance more than the cold adapted. In the transect, if you see in the left part of the screen, that was the first study that Ken Philip did with mice, looking at these migration rates in the Andes. And that was include only the Andean plots from the network. And the results were like, species were moving at the rate of 2.5 to 3.5 meters per year. And there was another study too in Colombia suggesting more or less the same rates in the Andes. And recently there was another study from the Philly lab led by Ken and Belén Fadrique, that they look how widespread and heterogeneous were those thermophilization rates across the Andes. That was a really interesting study published in Nature. But all those studies were focused in the Andes, and maybe have some caveats because most of the species, the ranges goes to the Amazon, and some of the Amazonian species goes to the Andes. So here in the x-axis, you have these black lines that represents to each species in the transect. And then you have elevation, which is the range. So you see that some Andean species extend the ranges down to the Amazon, and then some Amazonian species extend the range down to the Andes. The red line that you see are the mean or the center of the ranges. And you see that there is like around 20% of species that if you exclude just for the Andes, you will be excluding the populations in the lowland forest. So in this study, I include all those plots with all those species with the full range that we can include all the populations from each plot. And then we can see how those species are responding, because this is a continuous gradient, not gradient where you see these abrupt changes, because you have this continuous forest, and that's a really good advantage to work in the ABRAC transect. And another thing to consider is this approach when you look just individuals or basal area. So if you look the approach of just individuals, you see that tree 1, 2, and 3 are the same in the system. But if you weight it by basal area, you see that the tree 2 will cause more implications in the ecosystem function, for example, for carbon. And the results I will see is using this basal area weighted approach, where we include just the trees that we include the basal area for this analysis. So to calculate thermophilization, first was calculated the thermal migration rates. For that, we calculate the thermal optima, and this is following the protocol from Philly, from Ken Philly. So first, and this is an example, we have to download all the distribution from each species in each plot around the gradient from the GBIF, and I took all the records for more than 10 records. And you see that, for example, Califra cuniata have all these records on GBIF, the pink dots that you see, the pink pins. And then that you extract the temperatures for the warm climate, and then you get the mean, and that's the optimal for that species. And the average of the species in the plot are going to be the optimal for the plot. And that's going to be the community temperature index, is this thermal optima weighted by basal area. And the thermophilization rates are calculated as a net change of CTI, this community temperature index, over time. And then I will show you some analysis of like individual changes in the abundance for each species, this thermal migration rates for the most abundant species in the plot. And this is the criterion to have more than 50 individuals and then be present in more than 2 plots to see those shifts or changes across the plots. And first, this is what we have. We have, in all the graphs that you see now, elevation is going to be in the x-axis and the y-axis will differ. So here we have community temperature index against elevation. So each dot represent each plot. And again, the transition between the Amazon and the Andes, and the blue bar that is the cloud base. So you see that CTI is highly correlated with elevation. And this basically saying that temperature is a physical process and the plants can predict that almost perfectly. And we can ask them for that. For this analysis, we use all the plots even with one, we can use the plots with one census. We don't need the multiple census to calculate this community temperature index. And then we calculate the anomaly of this community temperature over time. And that was calculated as the absolute difference between the plot CTI in a year or in a given year and the plot initial CTI. You will see that each of these lines will represent each plot, and you will see the directionality and how variable it is for each plot. That you see that some of the plots have this directional change and some other plots have, a lot of variation, and some are positive and some are negative. So the plots have positive CTI, that's suggesting that an increase in abundance of those warm adapted taxa, which is consistent with the thermophilization hypothesis. And we have 38 plots with more than 2 censuses. All the plots with more than 2 censuses were included here. Then the thermophilization was calculated for each plot as the slope of a linear square regression between the CTI and the census. So we fit all these regressions to each plot and of the CTI and the census year, and we have those thermophilization rates. So here, each dot represent each plot again. Red colors represent positive thermophilization rates, and the blue colors, the negative thermophilization rates. And we have more positive than negative. And this, so the overall number of the plots with positive thermophilization was more than expected, under the null expectation of equal proportion of the plots with positive and negative thermophilization. And this was significant under this binomial probability. And again, these positive anomalies indicate increase of abundance of lowland taxa or warm adapted species, which is consistent with the thermophilization process. And if we see for which plots are significant, the red and blue are significant for each plot, and the gray are not significant, and that's still significant overall. And then we can fit, no parametric curve, and we see that around mid elevations is where we have these greater thermophilization rates. And the other thing to see here is that around the Andes, if you see just the Andes, there is a lot of variation. And around the Amazon, there is like less variation of thermophilization among the plots. Overall, so the migration is of 0.0020 degrees Celsius per year, which is equivalent to 0.43 meters per year. We can calculate this using the lapse rate of the transect, which is 5.5 degrees Celsius per 1,000 meters. And this is really low, as previous studies showed. So the trees are not moving, not even at 0.5 meter per year. And we look just for the Amazon and the Andes using this line, this arbitrary division. we see that the Amazon has this negative or close to zero thermophilization rates, and the Andes is positive, is greater than the Amazon, but still low. And if we put these results under the current warming, we will expect that the plots will be moving at this slope, whereas this red line. However, our plots or tree communities are moving really slow. So they should be moving around 9 meters per year, but they're moving less than a half meter per year, which is not really good news for the system, because most of these species could be facing extinction now. And now, how is this contribution of these different beetle rates of mortality, recruitment, and growth to this process of thermophilization, more or less to understand this mechanism of thermophilization? Here's an example that you will see. You will see that mortality is in black, recruitment is in gray, and growth is in light gray. And then you will have thermophilization for each plot. You will see these bars that indicate positive if it's red, or negative if it's blue. So here you see it's a little messy, but point your views in these black bars, and the black bars are driving these thermophilization rates. Around mid elevations are positive and high elevations are negative, but mortality is the one driving. And this is significant. We use this mixed model. However, those plots are not made just, the whole community, but they're made of single species. And if we look for a single species, this is what we found. So here, each dot represents to each species, and the error bars represent the multiple census. So we select all species with more than 50 individuals and be present in more than 2 plots, like I mentioned before. So we have an N of 79 species that fit that criterion along the transect. And we see that we have more positive than negative. And this is significant too under this binomial probability. And again, species in the Andes are more variable than the Amazon. And if you see this error bars, there is a lot of uncertainty of this process. It's still challenging to figure out exactly what's going on here. But the message is that at the species level, they are shifting to the distributions. And some of these species here in the lowlands, we have like Iriaria deltoidea, that the migration is positive. We can see to which of those species are, with the significant rates. Few of them are significant, the red colors, or the blue colors that are significant but negative, but most of them have this not significant rate, but still they are positive. And the overall rate is almost close to the plot level. It's around 0.44 meters per year, this thermal migration rate. Just a summary, so the trees are moving up, they're changing these distributions, but slower as we thought. And the Andean trees are changing the species composition or moving faster than the Amazonian trees. We see that mortality is the driver of this thermophilization over time. And one hypothesis is that at the species level, what's happening that is because of the short time for these long-lived organisms, we are observing a contraction range. And in the past, there was a migration of 0.2 to 0 meters per year. Currently, as a mean, there is like this migration of 0.4 meters per year, but they should be moving at 9 meters per year, according to the current warming rates. So it says that there is not equilibrium with the current or future rates of climate change. So those species are not in equilibrium, or tree communities. So now we saw these demographic rates, and we saw how those communities are shifting the composition over time. So the question is how that's affecting the ecosystem function, and we will see just the carbon dynamics. So we know that carbon is important, but the forests are important because it stores around 50%, 55% of the total carbon. But we need to keep that amount of carbon in these trees to mitigate global warming. And there is this study, one of the few that, all the tropics and the North tropics are these carbon sinks where they're storing a lot of carbon. And there is this tendency over time of storing carbon in those systems. But we need to test that. So to calculate, we use these standardized allometries to estimate carbon. But for the Andes, it was a little challenge because in the equation, we basically need wood density, diameter, and tree height, and use those constants from Chafe et al. But the problem is not with the allometry, it's with these three parameters, basically. First, because tree height in the Andes, like I mentioned before, the trees are not growing straight. They're like in the J shape. So there is a difference, and a significant difference between the length and the height of the tree, between the Andes and the Amazon. So for that, we measure with ipsometers almost all stems in the Aberg gradient, over 10,000 stems. And like I say, we observe the significant difference between the length and the height. And I can show you later if you have interest in see those graphs. It's really interesting. Then we face another challenge because there is no wood density data for the mountain systems. And wood density is an important proxy for the life history of the trees because of the mechanical support and these properties and these mechanisms of defense and this trade-off between growth and mortality. So there were no data set for like some timber species in the Andes. What we did is collect and get this comprehensive data of wood density in the transect. We collect in this elevational gradient for almost 900 individuals that are around 300 species. And we merge this database with the global wood density, and we assign to each species, to each individual, those values to calculate carbon. And this is what we found. So we have our 41 plots. Here we have above ground carbon in megagrams of carbon per hectare against elevation. Each circle represents each plot. And we see that there is a interesting, so these low values around here in the Amazon are the floodplain forests, and then these higher values close to 500 are the terra firma forests in the Amazon. So carbon, it goes a little bit up and then decreases towards the tree line. And there is a lot of variation too. And we don't see here this kind of abrupt transition in the cloud base, but we see just Amazon and Andes below the cloud base, we see that carbon can be kind of constant and then decreases to the tree line. But overall, it decreases from the Amazon to the tree line in the Andes. And how was that carbon net change over time? So here, each dot represents a plot again. The size is the number of censuses, and the red colors shows this positive carbon. So those plots are gaining carbon, and the red color suggests that those plots are losing carbon. So you see that overall, around the cloud base are where the plots are losing more carbon around the gradient. And in the Amazon, the plots are gaining more carbon. And we see that over time. We see that, so here, the blue colors, again, positive carbon change or positive carbon net change. The red color means this negative. The plots are losing carbon. Each dot represents a census of the plot over time. So since 1985 or 1983, I think was the first census. So the time here is 38 years. And the circles are then the Amazonian plots, and the triangles are the Andean plots. And something to consider too, that these bars, these yellow bars, horizontal bars, are the droughts that happened in the system in 2005, 10, and 16. So overall, there is a positive carbon change, and the Amazon are gaining more carbon than the Andes. But the tendency is decreasing over time. So for the Andean plots, it's this gray linear regression that you see here with the confidence intervals. So we have this negative slope, and so it's becoming from a sink of carbon to the source of carbon, and maybe those droughts play an important role in that. And for the Andes, sorry, for the Amazon, this dashed line is still positive, but the Andes, no. So in the Andes, we're losing more carbon. and the tendency is negative. As a summary, so there's a lot of carbon loss around the cloud base along the gradient. So the Andean Amazonian forests are still acting as a carbon sink, but it's a small net carbon sink. And overall, over time, so we observe this long-term decreasing in carbon accumulation over 38 years. Just to summarize everything, trees are dying. This is one of the main things that's happening in the transect. And those species perhaps are facing, or they are under selection. And those processes that we observe of changes in species composition or tree migration could be this result of this synergistic process between these changes in temperature and water stress. But also, we see that there is an idiosyncratic response of each taxon to warming and drought events, and we need to find out which species. And this transect is still acting as a carbon sink, but there is this long term of decreasing carbon accumulation over time. And just to finish, so this is what we did in the work, and now in my current position. So I'm working right now in this paper, looking at some of these questions in the Central Andes using the Bolivian data to the Madidi. But the overall goal of the study is working here at the Live on Earth Collaborative in collaboration with all of those different institutions. I'm working with Jonathan Myers and Sebastian Tello, and also, of course, working with all the ABERC members, is to try to generalize these results across all Andean and Amazonian plots from Venezuela to Argentina. To help and understand and contribute to answer this fundamental question of how tropical forests are responding to global change. And basically to test these three hypotheses, this species migration hypothesis in response to climate change, to test this biotic attrition and enrichment hypothesis, which basically suggests that lowland ecosystems are gaining species. Lowland ecosystems are losing species, and Andean ecosystems and mountain ecosystems are gaining more species due to climate change. And also to test this accelerate forest dynamic hypothesis that suggests over time, those forests are becoming more dynamic, but this is not tested in the Andes yet. Just to finish, one of the big thoughts is that tree migration is a canonical process as the climate shifts, and we need to share this detailed information for the decision makers. With that, I will say, apologize for being late, and thank you for your attention. I'm really open to answer any questions. Thank you so much.
Okay, thank you, William. If you stop sharing your screen, we can go back to this. So, yep. So I'll open the floor to questions. Anybody want to kick off with questions?
So William, thanks for a great talk. It was really interesting. I was struck when you were talking about the rates of migration of trees on the slope. The shape of your response curve was really dominated by 3 negative points very high on the slope in the Andes. And I'm just wondering if there is something going on close to tree line that is actually sort of influencing the statistics very strongly there.
So there is one hypothesis that these transition zones maybe are not permeable, and maybe these like environmental conditions are more strong influence the distribution of those plots or those species. And in this paper from Belen and Ken, they showed that the tree line as a transition zone and also the cloud base as a transition zone overall, they have these negative thermophilization rates. So there is something going on in those transition zones that we don't know yet.
I was just wondering if the history of those plots, there may have been people messing around in those plots up close to the tree line where there wouldn't be, perhaps down at 1,300 meters or so, and that you may be seeing some growth responses to historical land use there, or prehistoric land use.
Indeed. And also maybe the composition is going to be resilient to this land use. And you already have this resilient composition, and we are studying those like leftover trees that happened or they were not used to be there before. So it's interesting because in the Manu, where we all are working, so we did this like temporal 1 hectare plots just in the tree line. And there is some sites that the composition is really different. These isolated sites like the Apu Kanyahuay or the Apu Pitama. So the composition of those forests, of those patches are really different than the composition around Akanaco, which suggests that those are more resilient to land use change, maybe agriculture, cattle, and also fires. So those are more dominated by Clethra and Weinmannia, but the other sites are isolated are dominated by Polylepis and Genoxis and other Asteraceae. So there is for sure a history of those plots in the tree line, and there is for sure that land use is causing, or caused already some shifts, and maybe we are studying those resilient or remnant tree line forests in that area.
Yeah, thanks.
Okay, I'll follow up with a question. With this tree migration, maybe this is a question as much for Ken and his team as us for William. Do we see any evidence of functional traits, whether dispersal traits or physiological traits, that correlate with which species are being effective at migration and which species are not doing so well?
Yeah, so we didn't did that analysis yet, but we always talk about doing the functional traits. What I can say is that that growth, like fast growing species versus slow growing species, you can see a difference in the grade. So when I did the analysis at the species level, the fastest migrants are the fast growing species. And you have like not really successional, but fast growing in the plots. For example, we have some of the Laurasis was the fastest, Laurasis ocotea, and some of the Chloranthasis too. These like fast growing species are moving fast. So I don't know for the other functional traits like NLMA or some chemical leaf compounds or, other functional traits. Oh, with wood density, because growth is related with wood density, you can use wood density as a proxy. So this is so far what I observed, and I did. And I don't know if Ken has more insights about it. But another way maybe could be to see if those shifts are due. I mean, instead of looking for taxa, we can look those shifts for the function, right? For the traits.
So I don't have much to add to that, except, that's something that we've always wanted to do. And I think that's something, William has really led the charge with at the species level, so should be able to get some of those, linking the species migration rates with the functional traits along the Cosnipata transect. For the work in Colombia, we did assign we had Pablo Stevenson assign the dispersal vector for each species and try to see if there's differences between the dispersal vectors and their migration rates. And it was complicated and there wasn't much clear there. But overall, if we kind of just didn't think too much about the vectors, but just looked at things that had the potential to disperse far versus things that did not, there was a difference there, that things that could disperse far were migrating faster. So things that were dispersed by insects or that were dispersed by gravity, those were not migrating very fast, where things that were dispersed by birds or by mammals or by wind were migrating faster. But the pattern was relatively weak in part because, it's a very coarse assignment you know, to say mammal dispersed or bird dispersed. There's differences between mammals and birds, you know, which the species is doing them. So I think we don't have the great trait data, you know, and we haven't been able to match it up yet with the species yet, but hopefully William can get on that and figure some of that out.
Yeah.
Maybe, sorry.
No, please go ahead, Mark.
I was just going to say, it may also be worth looking at sexual mechanisms. You know, dioecious plants tend to disperse better than monoecious plants and hermaphroditics and so on. So that may be another set of traits to look at.
Yeah, so this is the question that we have. So I show you at the species level that we have winners, species that then do nothing, and losers. So the question is, okay, what makes a winner, tree a winner, right? And we can explore these functional traits. But then I show you that mortality is the big driver. So maybe to understand, not just including the functional traits, but to understand this process, we need to figure out why those trees are dying. And one of the hypotheses is that because of those multiple droughts, maybe they are dying by embolism or cavitation. And then the question then becomes, okay, so the trees are dying for this mechanism now, and this is the key question that I always talk with Ken and Miles, is where in the range those individuals of a single species are dying, because that's important. So we know the distribution of the species, the range of those individuals, and according to this migration, for them to shift the ranges, we have to expand the upper and contract the lower. So right now we are managing this hypothesis, and the data suggests this, that the species actually maybe just contracting the range and not really expanding, and we need to prove that. So if that's the case, so those species will be contracting the range and perhaps leading to extinction. But we always look at survivors, and these winners, we need to find out why they are the winners.
Well, I think we got to be careful.
It seems to be, sorry, go ahead, Ken.
You know, one thing to keep in mind is that things that are not migrating, they might actually be the winners because they might be more adaptable, you know, be able to adapt and acclimate. So they might actually have traits that allow them to acclimate or tolerate these conditions better, so they don't actually need to migrate. So that's a big question mark in all these things, right? You know, we're just assuming things need to move in lockstep with changing in temperature, but some things might not care about temperature at all. So, you know, there's a whole other set of suites of traits that might say, okay, these things aren't migrating because they're able to tolerate changes in their environment. Or we're doing a lot of work now looking at microenvironment like leaf temperature. Species might be able to control their leaf temperature, and so even as the air temperature warms, they might not feel it because they can keep their leaf surface temperatures the same. So you know, it becomes very complex because obviously it also matters on the life stage, you know, sapling and seedling and germination, dispersal, all these different things. But there's so many different ways that traits can influence the need for species to migrate and their ability to migrate, whether that's through contractions or expansions, that it becomes a really tough question to look at from, you know, you could probably do it for one species, but to think about for hundreds or thousands of species, it's kind of an intractable problem.
Are the mortality rates different for those that are migrating faster? So do you see any correlation between migration rate and mortality behavior?
Yes. Yes, Patrick, there is a correlation between the mortality rates and the species migrating faster.
So the faster migrators also have higher mortality rates?
Yes, and in some species, those fast-growing species, you can actually observe, but it's not significant or strong, this shift where they have more recruits in the upper range and dying a lot in the lower range. But for most of the species, they're just dying. That seems that's the primary mechanism.
And do you see that pattern across fast and slow growers as well? So you've got fast and slow growing and then fast and slow migration.
I didn't check for that one. That's the next step. I don't want to lie to you. Yes.
It's very interesting. It's a great, that was a great presentation, by the way, William. Really, really nice. Thank you.
Good to hear you, Patrick. From far, far away, I will guess.
Indeed.
Yeah. So just to follow on, it sounds to me it's critical to understand whether the range is contracting or expanding, whether thermophilization is a sign of weakness or a sign of resilience. It seems that you could interpret it either way, depending on which end of the range is shifting. The other question I had was around cloud base, where you seem to have lots of mortality going on. It could be due to past land use, etc. But it's also what we would expect if cloud bases were rising in response to warming. And obviously that would be a very different signal from thermal signals, because it's a very different set of traits that end up shaping that. And so what you're seeing possible biological evidence that there is some sort of shift happening at cloud base, and more as cloud forest species move out of cloud inundation into more submontane environments, we're seeing this sort of surge in mortality as they experience those different conditions.
No, from my understanding, in the transect, like there is not other organisms that are showing those shifts in the cloud base or not. But that's one hypothesis that we say about this cloud-based shift to the climate change. And those communities may be like the other cloud forest species. They take water from the leaves, and if you don't have clouds around, there's a big water stress, and then those trees just die by embolism. But alternatively, and around that cloud base, there is a high probability of landslides, like Catherine will show perhaps. And this high probability of landslides are related to high rainfall too. Seems that around the cloud base, there is a high turnover in species, where you have this heterogeneous species composition of fast growing and slow growing. Of course, that will depend on the size of the landslide and the frequency. But seems that there is a lot of turnover around that. So that coupled with the shift of the cloud perhaps are increasing the mortality of those communities.
Okay. So anybody else have a question? Okay, I'll put you in a slightly bigger picture one. It slightly links to your conclusion. So from what you know now, it's a two-level question. What do you predict the system will look like in 2050? And what are the big unknowns in you having confidence in your answer?
That's a hard one, Jasmine.
Yeah, but I think it's speculation, but I think it's useful to think about what have we learned in terms of what can we say about where the system is heading, and what do we need to know to have confidence in that? And can we know it by 2050, or do we have to just observe the system? Or do we actually have any predictive power in saying what's happening?
Yeah, what we'll see by 2050, I think, is that a shift in the species composition. We will see maybe, so in 30 years, even it's for a tree it's a short time, right? But I think we will observe those shifts, and if mortality will keep increasing, we'll have many gaps in the forest. However, what I see is that I think we need to really concentrate and figure out how or which species are doing better under these novel conditions, to use that for conservation purposes, and maybe dedicate more time of understanding life history of those species, do these reforestation plans to reforest these clear areas where we can use those species, knowing that they will be more resilient to climate change. Like putting the efforts to understanding more of those species, rather than it's important to plant trees, but it will be more useful to plant trees knowing that those trees can face better climate change.
Okay, great, William. Anybody have any final questions? if not, I think we'll send another round of applause to William for thank you for kicking off this seminar series. What I have done is record this, and if William's okay with that and others okay, what I'll do is I'll stick it up on YouTube and share it with everyone. And eventually, my hope is that we can build these seminars into a library of presentations that are more widely accessible to the people here on this call now. We'll try and do that. So we'll take a little bit of a break. What I suggest, if Mark's still there, is that the next seminar is led by Mark, although feel free to invite in other contributors, and looks at the human past and how it shaped the structure and composition of this valley. So megafaunal extinctions, but also perhaps if you have any thoughts or insights into past anthropogenic change, the history of the tree line with early human arrival, etc. And we focus on that. So great. So let's try and fix a date. So we could either do a month from now, which is late August, but if people feel that's still in the holiday season, we could defer into the first week of September and then kick off a regular series from early September. Any preferences?
Either works for me.
Okay, well, if people feel they'll be around in August, they won't be end of August, going to suggest. So,
27th?
We could aim for something around the 27th of August, if that works for most people on this call. Okay, so I'll fix that in the diary and send an email around everyone. And again, feel free to invite. I had a few requests from Josh and others to add extra people to this call, which I'll do. And feel free to suggest people that should be added to this mailing list if you feel your students or colleagues could benefit from this series of calls. So August 27th, with Mark Bush. But Mark, if you have suggestions of other contributors, feel free to think of other people who might want to pitch in little perspectives and ideas to that as well.
Sure. Okay. Thanks.
Great. And then we'll plan. We'll brainstorm a bit on subsequent seminars. We had a great list of volunteers early on in this thing, so I think we're set for the first 3 or 4. Okay. Thank you. I think they're really valuable. I'm really quite enjoying seeing everyone and then hearing everyone's thoughts and ideas and updates. Okay, so have a good summer break, everyone, and or winter break, depending on where you are, and see you on August the 27th. Okay.
Thanks, everyone. Thanks, William.
Ciao. Well done, William.